Good morning in the West and good afternoon here on the East Coast. I'm Bill Nierman, the Director of Infectious Diseases at the J. Craig Venter Institute. And today I want to talk to you about Aspergillus fumigatus, invasive aspergillosis, and some aspects of treating invasive aspergillosis with azole drugs. So first, looking at this disease and how it's really dependent on the host status. So Jean-Paul Leger described Aspergillus fumigatus as really a grass eater. It's not a pathogen. It's ubiquitous everywhere on the planet practically. It's in our homes, it's in our lawns, it's on the walls of our offices. So we daily breathe in several hundred spores, the so-called conidia from Aspergillus fumigatus, but we don't come down with the disease because of the fact that under our normal immune system, the alveolar macrophage actually are very effective at killing Aspergillus fumigatus spores. It's only under cases of immune dysfunction where you actually get invasive aspergillosis in either subacute or acute stages. And so it's actually relatively recently in human history that invasive aspergillosis has been something that we've even noticed. The reasons for it are something I'll talk about in a bit. But then on the other end of the immune status scale is immune hyperreactivity in individuals such as those with severe asthma. Under those circumstances, the regular exposure to Aspergillus spores results in colonization that can really lead to problems where you've got fungi colonizing the sinuses and causing this allergic bronchopulmonary aspergillosis. So that, you know, in normal immune system, there are these other conditions that are listed here, but these are really low incidence circumstances that in fact we are in normal immune status very effective at dealing with the fungal spores that we encounter in our daily lives. So in the cases of immune-compromised folks, the leading cause of mortality is invasive aspergillosis for these infectious mortality for these folks. It's increased in incidence simply due to the fact that our medical successes have given us a longer life expectancy. So we actually are an aging population relative to human historical time. And the additional component that causes this increased incidence is the fact that our medical technologies, particularly tissue transplant and chemotherapy for treating cancer, leads to immune suppression. And the immune suppression makes us vulnerable to infections by these spores that it's very difficult to avoid. The mortality for invasive aspergillosis is between 50 and 80% for a couple of reasons. One of the reasons is that it's really fairly difficult to diagnose, and so typical diagnosis comes in after the infection is really pretty well established. And the other issue is, which is going to be something that's going to be a major part of the talk, is that the drugs that are used for trying to manage invasive aspergillosis patients really are not very efficacious. And so in the absence of these really effective drugs and the late diagnosis, we see mortalities at these levels. So I indicated medical transplant technologies contributing to these diseases. A group of diseases that are genetic, the innate immune deficiencies, also are exposed to higher risk. In particular, chronic granulomatosis disease has a high lifetime incidence. Also, lung transplantation in particular has a high lifetime risk. But most of the people that get this are bone marrow transplants or neutropenic patients that are undergoing chemotherapy. So having looked at the human side, let's now look at the fungus, the Aspergillus agents that actually cause these diseases. The image here is of Aspergillus fumigatus. You can see the filaments on the colony on the left, and you can see that gray-green color. That's the spore color, the spore coat pigment, and you see these microscopic images of these fruiting bodies in the fungi that produce these spores. In particular, for Aspergillus fumigatus, the spore size is really small compared to other Aspergillus species, and this certainly facilitates it being dispersed by air. And also, when we breathe these things in, the small size really results in deep penetration into the lower respiratory tract, which probably contributes to disease. From a clinical presentation, you see opacities in the regions of the lungs in boxes A and B, and so where you see indicated by the circles and the arrows are areas of high fungal burden. By CT scan, you can see these concentrated fungal infection sites indicated by the arrows in C, and in D you actually can see a consolidation of a portion of the lung that's resulted by the severe tissue damage that's resulted from this fungal infection. The neutrophil ingression is shown in E. The fungals having invaded the lung tissue really causes recruitment of the neutrophils, which actually causes a lot of the damage in the case of invasive aspergillosis. And you can see in panel F, by staining for the fungus, you can see the filaments actually within the lung tissue. Light microscopy, the lesions are clear. You can actually see the filaments by staining them with something like calcofluor white, which makes them readily apparent within these lesions. So I've been talking about Aspergillus fumigatus. Aspergillus fumigatus, for most of the world, is the leading cause of invasive aspergillosis, but the fact of the matter is there's something like 200 Aspergillus species that have been identified. It's noteworthy to point out that only about 7 of these species have been reported to have caused invasive disease. The second most prevalent Aspergillus that causes this disease is Aspergillus flavus. And there's actually parts of the world where Aspergillus flavus is the primary, the major cause of invasive aspergillosis. What I'm showing you here is a very recent report by our collaborators Janice Suji and Jun-Kwan Chung, where they identified a new species of pathogenic Aspergillus, which they named tanneri. It's only been known to have killed 2 patients so far. They are at the clinical center. There's a large respiratory service at the clinical center at the NIH campus in Bethesda. And so the severe cases of particularly the GCD patients get referred to the clinical center when they're really having difficulty managing them in their home medical facilities. A particular attribute of this new fungus is that it's resistant to every known antifungal that's used for managing the disease, and so for 2 diagnosed infections, both led to the death of the patients. Now looking at a project, because as I indicated, there's more than one fungus that can cause invasive aspergillosis. So here I want to go to talking about how do you identify virulence factors in these, and so I'm going to just give you some examples. Aspergillus tanneri really has given us an opportunity to see different views of virulence. So here is shown 3 models of these are animal models of invasive aspergillosis. The upper panel A uses wax moth larva, Galleria melanella. We use this in our lab. It's a really inexpensive approach to doing pre-vertebrate animal virulence testing at much less cost, and a lot of the findings that are noted in this system actually carry over to showing virulence in the mouse models. So the green is the percent survival for those infected with Aspergillus flavus. In this model, Aspergillus flavus certainly is the obvious most virulent of these 3 species. The tanneri in blue is the intermediate, and Aspergillus fumigatus is actually the least virulent in this model. When you drop down to the Balb C mice, panel B, this mouse model uses cortisone suppression, and the way this is set up using the immunosuppression is to try to mimic as much as possible the mouse model the suppression conditions under tissue transplant or bone marrow transplants in humans. And in this model, you see that all 3 Aspergillus species seem to be exhibiting fairly similar patterns of percent survival in this experiment. And then GCD mice, now in GCD, these mice, the innate immune cells do not have the ability to generate reactive oxygen. And so as a consequence of this, they're a bit defective in the ability to kill microbes. And humans with CGD are particularly susceptible, as you saw in an earlier slide, to invasive aspergillosis. And in this model, Aspergillus fumigatus is the more severe pathogen, while the other 2 really are exhibiting very little killing over the course of this experiment. So the 3 models here really just give different profiles for these pathogenic Aspergilli. Here has a taxonomic tree of these 3 Aspergilli shown here, fumigatus, tanneri, flavus highlighted. Within this taxonomic tree are lots of species of Aspergillus that never appear in clinical cases. And so there's something about these 3 that make them really different than their other members of the genus. All of these organisms essentially compete in the same soil environment. They've all established the ability to compete successfully in this environment, and since the human infection is really a dead end for this fungus, it's not a normal part of their life cycles. They've all developed the capabilities of surviving in an immune-suppressed human as a consequence of the selection they've experienced in the soil. And the obvious example that people cite in talking about this virulence traits acquired in the soil environment is that undoubtedly these fungi regularly encounter amoebae and in the soil, and the ability to survive an amoebae from the perspective of the fungus requires pretty much the same skill set as to survive in human macrophage, presumably. Just as an aside, we've done transcriptome analysis with Elaine Bignell in the UK at Imperial College in London with flavus and fumigatus, and both of these fungi in mouse lungs really increase the expression of sets of clusters of genes that Elaine has labeled as virulence clusters, because presumably these things are induced in the stress of the animal lung environment to allow survival there. But of note, a separate observation we made in our group with Natalie Fedorova contributing to this is that the clusters, these clusters are in accessory regions of the genome. These are genes that are not dispersed throughout the Aspergillus genus, but are fairly unique to the species. And so clusters of genes are upexpressed in mouse lungs in fumigatus. They're upexpressed in mouse lungs in flavus. But the fact is that these clusters of genes have very little sequence relatedness between these two species. And so our hypothesis here is that these fungi meet the same sorts of challenges in the mouse environment, but they use different tools that they've acquired from their environmental exposure, environmental selection, to be able to survive here. And so we're going to do an exhaustive comparative analysis between flavus, tanneri, and fumigatus to see if we can really come up with some understanding of the range of functions within these fungi that are essential for establishing these invasive infections within humans, immune-suppressed humans. So, getting on to more what I'm trying to make the key components of this presentation is how we manage invasive aspergillosis patients in the clinic. Everybody is well aware of the crisis in infectious diseases that is maturing due to drug resistance in bacterial antibiotics. But compared to the bacterial problem, it's much more severe in the fungi. Bacteria have many more classes of targets because bacteria are prokaryotes and the patients, the humans, are eukaryotes, and there's a lot of metabolic and structural differences between the prokaryotes and the eukaryotes. This is not true for the fungi. Fungi are eukaryotes just as we are, and there's actually, in looking specifically at drug targets for treating invasive aspergillosis, there's only 3 classes of drugs, and they're targeting only 2 differences between humans and fungi. So the polyenes and the azoles take advantage of the fact that in fungal cell membranes, the sterol is ergosterol, whereas in human cells, the sterol is cholesterol. And so this difference, this relatively subtle difference, is actually allowing for these 2 classes of drugs. The ergosterol, the polyenes actually target the ergosterol in the membrane, and the azoles inhibit the production of ergosterol. The third category, echinocandins, take advantage of the fact that fungi have a cell wall and human cells do not, and so this inhibits a critical enzyme in fungal cell wall biosynthesis. So, just again in diagram here, the polyenes, specifically for treating invasive aspergillosis, amphotericin B, binds to the ergosterol in the membranes, and the azoles, several azoles that have been developed for treating the disease, and the azoles are the primary drug for treating invasive aspergillosis, inhibit the biosynthetic pathway of synthesis of ergosterol. And so here is the cell membrane diagram, and the cell wall of fungi is in 3 layers. There's mannan on the outside, there's the glucan intermediate layer, and then the inner layer is chitin next to the cell membrane. The enzyme inhibited by the echinocandins is the beta-1,3 glucan synthase, which actually polymerizes the sugars into making the beta-1,3 glycan of the cell wall structure. So amphotericin B is actually produced as a natural product by Streptomyces. It binds to sterols, as mentioned, and actually creates pores in the membrane that result in electrolyte and small molecule leakage that causes the cells to die. Echinocandins are cyclic lipopeptide antibiotics. They, as I indicated, bind to the beta-1,3 glucan synthase. They're analogous to the beta-lactam antibiotics for treating bacterial diseases, bacterial infections, because they cause a defective cell wall to be made, which results in osmotic fragility and cell lysis. But the primary, in showing where the glucan is synthesized in the elongating filaments of the fungus, fungal filaments grow from the apical end, and you can see by the staining of in panel A with antiserum to the enzyme, the glucan synthase, that it's concentrated at the end of the filament. By staining specifically for the beta-1,3 glucan, you can also see that the most abundant location of the glucan is in the growing apical tip. But the major drugs that are used for treating invasive aspergillosis are the azoles, and these are, well, what's shown here, circled in right, this ketoconazole is a class of azoles called imidazoles. They have this active imidazole ring that contains 2 nitrogens and a saturated 5-member aromatic ring. And circled in red is the triazoles that have the 3 nitrogens within the 5-membered ring. These azoles inhibit the enzyme the gene name is ERG11. It's an enzyme that actually takes lanosterol, an early sterol in the biosynthetic pathway to ergosterol, and performs an additional step in taking it to the ergosterol. So it's a cytochrome P450. The enzyme is a sterol 14-alpha demethylase, and it's responsible for this conversion. And the azoles actually bind to this enzyme, and there's been structural studies performed on how these azole compounds fit into the active site of this enzyme. So looking first at, I'm shifting into discussing drug resistance now, and so here's across the top is these E-strip for E-tests for drug resistance, where you grow the fungi on plates. You lay the strip on. As you go down the strip, you have increasing concentrations of the antifungal, and this diffuses into the medium, and it's calibrated using other tests to allow visual reading of the minimum inhibitory concentration of the drug on the fungus. And so you see Aspergillus fumigatus, the first sequence strain AF293, is sensitive to both voriconazole and amphotericin B. And then in the adjacent panels are the 2 isolates that killed the 2 patients of Aspergillus tanneri. You can see coloration difference around the strips, but in fact there's the colonies, the fungi grow right up against the strip. So these have resistance completely to the range of these test strips. And this is the problem in many patients, because the azoles are relatively ineffective, and the result of that is the need for long-term maintenance of patients on azoles. The fungus develops resistance over time. So in this diagram, you see the two sort of classes of triazole drugs. The A and Bs, the posaconazole and itraconazole, have these long chains. You can see the triazole rings there in the lower parts of the left end of these molecules, and those look quite different than the simpler compounds, voriconazole as C and fluconazole as D. So I indicated that structural studies have been done with crystallized protein and crystallization of the protein in the presence of the azoles, and the colored balls. So on the left is posaconazole, which has that long tail, the long tail on the azole compound, which I've circled in red. The one on the right is voriconazole, is in the same protein. The diagram shows 3 sites where sequence alterations at these sites confer resistance to some or all azoles. Also, you see that green structure. That's a heme molecule, and the triazole the triazole ring shown in the voriconazole panel B is sitting on top of the iron, really, which inhibits the oxidation reduction capabilities of the heme as a cofactor for this compound. And so you can see how this G54 in panel A is in very close proximity to this tail, and alterations with other, G is for glycine, and when something other than glycine is at this site, you do get resistance to the posaconazole. But not with voriconazole, and you can see that the G54 site, the glycine 54 site, is actually quite removed in the absence of that tail on the azole. So in this way, you can see that some alterations in this ERG11, also called CYP51 sterol demethylase, confers resistance to only some of the azoles, or it confers resistance to all of the azoles. So this glycine alteration at position 448 confers resistance to all of the azoles. So there's a lot of sequencing that's been done on resistant strains. A lot of positions have been mutated and been shown to contribute to resistance. In addition, there's tandem duplications in the promoter of this gene that results in overexpression. This has never been observed in clinical isolates, but it has been observed in Aspergillus fumigatus harvested from the environment. And one of the confounding things of all this, while the conventional wisdom is that CYP51A, the ERG11 gene, this demethylase, is the target of the azoles. There's a lot of strains out there that are resistant to azoles for which there's no mutations in this gene that can be used to account for this resistance. So is this resistance important? Well, as I indicated, many patients are on long-term azole therapy because of the need for treating for such a long time to clear the infection. And so in the course of long-duration azole treatment, the organism goes undergoes replication and mutations, and the selection and presence of the drug really selects for drug-resistant strains. So in invasive aspergillosis, mortality when the strain that's isolated is sensitive is the 30 to 50% I indicated earlier. But once you convert to a resistant strain, or if you're initially infected with a hospital outbreak from a resistant strain that's gone through multiple patients, the mortality progresses. Mortality is now 88%. So this is a dramatic difference in prognosis based on the resistant status to azoles by the fungus. And as I indicated, in the Manchester group that we collaborate with David Denning, where they've got a sort of a national center for invasive aspergillosis, 10 to 50% of their isolates from patients that are resistant to azoles do not carry alterations in this gene. So it appears from on the surface that resistance to azoles is much more complicated than a single target. Because of this, we've launched a study in collaboration with the Manchester group and in collaboration with Jun Quan Chung at the Clinical Center to really apply a multi-faceted approach to identifying resistance modulating genes in the genome of Aspergillus fumigatus. And really one of the things we want to accomplish in this study, because there's so few drugs that can even be applied to treat this disease, is to identify candidate drug targets or co-drug targets for subsequent drug development to really offer some greater opportunity for actually managing patients with this disease. And so we're using 4 different approaches. It's really just 3 approaches, with the last confirming that the alterations we find actually have relevance for clinical isolates, because we've seen the same alterations in those clinical isolates. So the A approach is transposon sequencing, Tn-Seq. All of these approaches use next generation high throughput sequencing as the key technology for determining the findings. So Tn-Seq is probably the most powerful of the 3 approaches here. Tn-Seq has the ability to look at every gene in the genome and evaluate its fitness under some selective pressures. And of course, the selective pressures we want to impose here are stresses from azole drugs. And so it employs a large library of clones of Aspergillus fumigatus. Each member of the library is a separate clone with a single transposon inserted in a gene, causing a loss of function of that gene in that particular individual member of the library. And then the library is passaged under selective pressure. We put some level of azoles for passage conditions. And then the relative abundance of each member of the library is determined before and after the passage. And because a clone that is more resistant to the azole on passage is going to grow faster than one that is sensitive, by measuring the relative abundance of each of the clones with transposons in a single gene each, you can actually calculate the fitness contribution of that gene under those passage conditions. Don't worry about the details of this slide. The stuff, this is a bacterial Tn-Seq process. It actually turns out being a bit more complicated in Aspergillus fumigatus for what we did, and so this was a much easier way to say it. Just be aware that the first step, A, is making your library by putting a transposon into a large number of your fungal spores, fungal clones. And so that in such a fashion that each of the resultant individuals has a transposon that's at a unique site in a gene or in an intergenic region within the fungus. And this is your Tn-Seq library. Then you take your library in line B, passage it through azole in vitro passage conditions that's going to put pressure on the members of the library. Most of the genes aren't going to affect the survivability or the growth rate in the drug, and so their abundance is not going to change. Some genes, when there has a transposon resulting in a loss of function, are going to make that individual member of the library a better competitive if it's actually making it less sensitive to the drug, or less competitive if it's making more sensitive to the drug. And so this gives you a relative measure of the fitness contribution of that gene through this process. And the way you evaluate that is by, in line C, going through a procedure totally different than the one illustrated here, which captures specifically pieces of DNA adjacent to the site of the insertion, so that you can actually identify what gene the transposon is inserted in that strain. And then these captured regions of genomic DNA adjacent to the transposon are then sequencing using an Illumina sequencer, and the sequence reads are mapped back to the genome, and each read reflects an individual in the library that was present in either the before condition or after passage, or both. And you can actually, by looking at the numbers of reads before and after for a particular gene, get a measure of the fitness of that gene under the selection pressure of being passaged in the Azole drug. So our collaborators at the University of Manchester have made the library. The gene count in Aspergillus fumigatus is about 10,000. There's about 10,000 genes in Aspergillus fumigatus. And they made a very large library that contains 500,000 individual clones, each of them having a different insertion site for the transposon. So this is very good coverage of the genome. Then the library was passaged by them in 16 times the minimum inhibitory concentration of itraconazole, and then it was analyzed by relative abundance of the clones before and after passage. And this analysis revealed actually 387 genes with strongly altered fitness. That is, these 387 genes either made the clone containing them in the untransposed state. These are the functional versions of the gene, either increase resistance to azoles, this azole itraconazole, or they decrease it. So it makes it more sensitive or more resistant. And so it actually plays in the response of the organism, of the fungus, to the challenge. Some of these genes, of course, it identified the CYP51A, that's the lanosterol demethylase, but there's also a lot of other kinds of genes that seem to be important for azole survival or sensitivity, including transcriptional regulators, tRNA modifying enzymes, just a whole lot of things, and then a lot of genes of unknown function. So this is telling us that it makes sense that there's a lot of strains coming out of patients that are resistant to azoles that do not have a mutation in the CYP51A gene, because there's a lot of genes in the genome of this organism that acts on the sensitivity or resistance to azoles. So that was our first strategy. The second strategy was simply taking two strains of Aspergillus fumigatus that were nearly isogenic, and that'll be important in a minute when we talk about the next strategy, and passaging them first round simply at a sub-inhibitory concentration of 4 azoles. We did this with 4 different azoles, and then we passaged them at 4x MIC, 4x for the minimum inhibitory concentration, in step 4, and pick 4 individual clones from each of the passage conditions. I mean, we picked 3, I'm sorry, it's 3 is shown there. So we picked 3 individual clones. And then we passaged those through a second round of drug challenge, this time 8 times the minimum inhibitory concentration. And then for each of those, we picked another 3. And so we ended up with, I think, something like 32 different isolates from 2 different levels of drug resistance, and we sequenced the genomes of all of these and looked for changes relative to the drug-sensitive initial parent. And so we got lots of variations, and so I'll put this together in a minute. But we used the very highly resistant parents of both mating types in a sexual cross experiment where we took 3 different resistant isolates that had been through this selection and crossed them with the other mating type starting sensitive parent. And so it's only been in the last several years that you've been able to have in vitro conditions for having sexual crosses within Aspergillus fumigatus. And Jun-Kwan Chung and Janice Sergi developed conditions that make this much more efficient than the earlier reports that require something like 6 months for a single cross. And so in this experiment, we did these crosses with parents from all of the different azoles that we did in the previous selection experiments in vitro, and then did the crosses, and then collected the ascospores. Now, the ascospores are the sexual progeny of the cross, and we passaged the pool of ascospores under 3 conditions: no drug selection, and then 4x MIC, and then 8x MIC. And the no selection should give us equal representation of the variations in the genomes because there's no drug pressure at all, and so that's a nice control that the crossing went as expected. And then at the intermediate and high levels, we expected to see enrichments for all of the genetic variations, all of the SNPs, the genetic alleles coming from the resistant strain, enriched in these populations. And then our last approach with the clinical center at the University of Manchester in the UK with David Denning and Paul Boyer, they sent us 36 total clinical isolates. 16 of these had azole resistance and 28 were sensitive. And some of these 16 resistant ones were sequential isolates that had different levels of azole resistance from the same patient. So we could actually see the progression of resistance within the same patient in some of these. And then we sequenced all of these and then identified SNPs, single nucleotide polymorphic sites, basically sequence differences across all of these. So some of these isolates had no CYP51A resistance, which was consistent with what we'd seen before. And I had mentioned that some of these were consecutive isolates, but one thing I want to indicate is that these are unrelated strains, and unrelated strains have a high background of numbers of SNPs between them. So what we wanted to do with this set of isolates is to look back at the things we found through processes A, B, and C and see if we see the same mutations coming up in the clinical isolates, because you can make the case that everything we've done in A, B, and C are only in vitro manipulation of the organism, and it's conceivable that it could be artifacts. So we really wanted to use these sequencing of clinical isolates just as a validation that we see the same kinds of variation within the clinical population as we see in our in vitro analysis. This is just a quick look at some of the enzymes in the ergosterol biosynthetic pathway from these clinical isolates. And the red strain and the green strain are resistant and sensitive, and the base change in the DNA sequence is shown at the very end of the middle column. There's a lot of differences between these strains. I think the number of SNPs between any two unrelated Aspergillus fumigatus strain is something like 70 or 80,000 SNPs. So this is not a very robust way of finding resistance genes, but it is a good way of seeing what is present in clinical isolates. And so this is sort of a summary table of condensed from a much huger data set. So what's shown here is the mutations that led to drug resistance using our various approaches. And so, as I indicated, the Tn-Seq, which is the third column over, identified two genes within the ergosterol pathway as essential. One is this ERG11, the CYP51A that I've talked most about. Essential meaning that when you put a transposon in this gene, that's a lethal event for that individual clone. So we never saw transposons in those, and so that's how we knew it was essential. This HMG-CoA reductase, hydroxymethylglutarol-CoA reductase, is an interesting story because it was noted some time ago that statins are drugs for reducing cholesterol in human. They come from Aspergillus and other filamentous fungi, but statins inhibit human hydroxy. This is one of the early enzymes in the synthesis of cholesterol, and so these are using to minimize cholesterol. And so presumably this gene, which we found mutated in all of our approaches, is also modulates the resistance to the azoles. And then you see these other things. Here you see scores for both things that contribute to resistance and contribute to sensitivity through some of these other factors. And actually the list is much, much larger than this. There's validation across all three of these columns, so that we're seeing the same mutations at some level using all of the approaches, although Tn-Seq really gives us a very high-resolution view of the role or the quantitative role of gene playing in drug resistance. So what we have now is a very large set of drugs, targets that can serve as targets for drugs or co-drugs, and which we've identified using multiple technical approaches here, which I think the approach makes it very robust. We've certainly seen, based on the initial observations, that a lot of azole resistance isn't resulting from CYP51A mutations, but is azole resistance actually is broadly polygenic, with lots of genes contributing to resistance or sensitive phenotypes. And even within the CYP51A, particularly with the two different classes of azoles, there's multiple alterations within this gene that leads to resistance. There's an even larger number of alterations which has no effect on resistance. This is really a gene within the genome which has a very high rate of mutation on propagation. I don't know if this is an adaptation to evolutionary pressure from azoles in the soil, but certainly compared to most places in the Aspergillus fumigatus genome, we see a much greater variation, even in the absence of any drug considerations within the CYP51A gene than most of the other genes in the genome. So with that, I want to credit the University of Manchester group, Paul Boyer and David Denning, and the NIH Clinical Center, Janice Suji and Jun Quan Chung, for the major contributions they made to this project. And within our group at JCVI, Stephanie Mona worked with me in developing the drug-resistant in vitro selected isolates, and Liliana Losada has headed a project here in collaboration with Paul and David, sequencing these clinical isolates in projects that were funded by NIAID. So with that, the presentation is concluded and open for any questions. So the first question is asking for a little bit more information on how the Tn-Seq works, because I guess it wasn't completely clear the way I described it. So the key is that with Tn-Seq you have a very large library that contains transposons inserted in virtually every gene in the genome in some of the clones within that library. So the library is a set of clones that have been transposed to put loss-of-function transposons into genes within individual members of the population. And then it's just simply a competitive growth assay through the selective conditions. So you have your initial library that you passage under drug selection, and under the drug selection, the members of the library which are more resistant will grow faster, and you'll have a larger abundance of those clones after passage. And you'll see those more often because you'll actually count the numbers of individuals in the library that have a transposon in the gene at issue. So if a transposon is knocking out a gene that actually, in the absence of that function, it's more resistant, they'll grow faster. And the inverse is, of course, true, that if you knock out a gene that's essential for resistance at that level, it'll grow more slowly, and so you'll see its population within the library decline. And so you'll see those as scoring fewer members after passage than before. And so it's really, if you adjust the numbers of cycles so that you don't get too many really maladapted insertions totally eliminated from the population, you'll get a very powerful measure of the fitness contribution of the genes under the passage conditions, which will give you a very broad view of all the genes that are playing in the phenotype. And the phenotype, of course, is resistance or sensitivity to the drugs, and those that play in the resistance. If they're druggable, then they can become candidates for drug development projects. So I don't see any other questions. Anybody have any last-minute questions you'd like to ask? So if there's no further questions, I'd just like to mention that you will receive subsequent information if you are interested in the continuing education credits. And with that, I thank you for your attention, and I appreciate the opportunity to really give you this lecture. Thank you very much.